Welcome back to our lecture on embryogenesis. This lecture will be highlighting week 2 of development, otherwise referred to as the week of twos. It receives this name because of the further differentiation of the inner and outer cell masses into its two layers, as well as the formation of two cavities that is also going to occur during this week. More specifically, we can say that the embryoblast, or the inner cell mass from week 1, further differentiates into its two germ layers, the epiblast and hypoblast, and in doing so creates a bilaminar embryonic disc. The trophoblast, or outer cell mass from week 1, further differentiates into its two tissue types, the cytotrophoblast and the syncytiotrophoblast, as part of initiation of the placenta. And lastly, the amniotic cavity and the yolk sac will also be formed. If you recall, we left week 1 with a differentiation of the outer cell mass into the cytotrophoblast and the syncytiotrophoblast. The inner cell mass, or the embryoblast, was also differentiating into an epiblast layer and a hypoblast layer. As the blastocyst transitions from week 1 into week 2, the syncytiotrophoblast cells quickly expand and further erode their way into the endometrium. As the erosion occurs, the blastocyst follows its way into the newly created space. This process continues until the embryo is completely embedded within the uterine wall by about 10 to 12 days post-fertilization. At the same time that implantation is progressing, the two germ layers of the bilaminar disc, i.e. the epiblast and the hypoblast, continue to proliferate extensively. As they do so, their cells begin to spread to form the initial development of two cavities. A day later, we see that the epiblast cells have proliferated so extensively that they have circled back on themselves. To create a small fluid-filled cavity known as the amniotic cavity. Further differentiation of these epiblast cells also take place. The epiblast cells that remain in contact with the hypoblast are destined to become the embryo proper, but the cells that define the roof of the amniotic cavity are not going to form body tissues. Instead, they are transformed into amnioblasts, which form the lining or amnion of the amniotic cavity. As a side note, the amnion is what secretes amniotic fluid into the amniotic cavity. Now, as the amniotic cavity forms, cells of the hypoblast also proliferate extensively and spread out along the inner surface of the cytotrophoblast to eventually line its entire surface. The space that is being defined by the cells of the hypoblast is the primary yolk sac. This sequence of images depict progressive development of the hypoblast cells as they proliferate and spread out along the inside of the cytotrophoblast cells. Once the cytotrophoblast is completely lined, the primary yolk sac subsequently gives rise to an additional population of cells, which are the extraembryonic mesoderm. These cells spread to surround the primary yolk sac and amniotic cavity. and separate them from the cells of the cytotrophoblast. You'll hear of these extraembryonic mesodermal cells later again when we talk about erythropoiesis in a future video. Once the extraembryonic mesoderm has completely surrounded the primary yolk sac and amniotic cavity, a large space develops within this newly deposited layer, known as the chorionic cavity. While important to embryonic development, the chorionic cavity is a transient space that will eventually be pushed aside by the amniotic cavity and disappear. At the end of week 2, the chorionic cavity has enlarged substantially. Notice that it now surrounds much of the bilaminar embryo and the two cavities associated with it, i.e. the amniotic cavity and the yolk sac. At this stage of development, the only attachment between the embryo and the cytotrophoblast is via a narrow strip of extraembryonic mesoderm. This area of attachment is referred to as the connecting stalk. At this point, the growing conceptus is in need of a blood supply and achieves this through the establishment of uteroplacental circulation. Throughout week 2, the syncytial trophoblast continues its invasion of the endometrium. As it does so, small vacuoles or spaces appear within it. These vacuoles eventually fuse to form larger lacunae. In the process of forming the lacunae, endometrial blood cells and uterine glands are also invaded. As a result, the lacunae quickly become filled with a mixture of maternal blood from ruptured endometrial arteries and glycogen from ruptured uterine glands. Uteroplacental circulation is thus established in time for the embryo to transition into week 3 of development. This concludes our lecture on week 2.